introduction. Hi, I am Hazwani from Advanced Medical and Dental Institute, University of Science, Malaysia. So today I'll be presenting my research title, Cytotoxicity of Silver Nanoparticles Synthesized by Catharanthus roseus aqueous extract on JCAT and HT29 cancer cell lines. So this is a very brief background of mine. I did my PhD starting from 2008 until 2012 that involved the effects of catharanthus roseus aqueous extract on JCAT cells and normal peripheral blood mononuclear cells. And it's kind of interesting to find out that, this extract has significantly inhibited the proliferation of the cancer cells, particularly the JCAT cells, which is human leukemic T cells, but on the other hand, it induced the proliferation of the normal peripheral blood mononuclear cells. So by the way, my field is cancer immunology. And then if you see the symbol, which is the National Cancer Council, so the symbol itself represents the flower of the catharanthus roseus. So my project was partly funded by this organization, which is NGO in Malaysia. So I decided to continue studying about this plant after I finished my PhD until now, but decided to use a different form of, instead of using a crude aqueous extract, so I decided to use nanoparticles synthesized by this, the same plant. So this is some of the few data of my PhD. So we did a gene expression profiling of catharanthus roseus treated JCAT cells, and this diagram represents the effects particularly on the apoptosis. There are two mechanisms, basically intrinsic and extrinsic pathway. And we, in the box, the red and the green one are the genes that were either up or down regulated. And the box with sign, with a star sign indicates that the pro-apoptosis genes, which correlates with our functional assays. Right? So in this case, we found that the genes involved were TRAF4, CDKN1C, GFER, and BIRC8. And this is the gene expression profiling of catharanthus roseus treated JCAT cells related with the cell cycle analysis and the genes that are most likely involved in our studies and correlate with our functional assays with LTBP1 as well as the RBN1. Because our functional assays stated that the aqueous extract has, arrested the S phase of the cell cycle. So for the introduction for today, I would like to, introduce a bit about the nano-oncology. So it can be defined as a new research knowledge that has potential to be used for the detection, target, and treatment of cancer. And nanoscale treatment itself is advantageous as compared to the micro-sized anti-cancer drugs, since it is more stable and specific, and requires only limited dose of frequency. So this might be due to the unique characteristics of the silver nanoparticles, or of the nanoparticles itself, such as high surface area per volume, increased porosity and solubility, increased bioavailability, as well as the different structural properties. Okay, so this will make the cells easily pass through the cellular barrier and can strongly interact with the cellular biomolecules. So all these characteristics can overcome the limitation of conventional cancer therapy, such as the less effectiveness of the anti-cancer drugs to reach the targeted sites. And a report provided in 2011 has summarized the nanoparticles-derived drugs in different phases of clinical development, and one of them is Abraxane, which is the albumin-bound paclitaxel particles. And this drug has been approved by FDA to be used for the treatment of non-small cell lung cancer and breast cancer. So there are many types of nanoparticles, I believe. In fact, I was exposed with many kinds of nanoparticles in the Congress, but one of them is silver nanoparticles, which is my major focus here. So it can be defined as nanoscale, matter size within 1 to 100 nanometers. Their unique properties include good in conductivity, stability, catalytic, and antibacterial. And they have been widely used in biomedical application that include anti-inflammatory, antioxidant, antimicrobial, as well as in medical devices. However, their potential application as anti-cancer agents are still new and remain to be investigated. Interesting findings have reported on its biocompatibility to healthy cells, but on the other hand, inhibitory effects on various human cancer cell lines such as glioblastoma cells, Dalton's lymphomacytis, cervical carcinoma, breast carcinoma MCF7 cells, HeLa cells, and lung cancer A549 cells. So the three major methods of silver nanoparticle synthesis include physical, such as thermal and laser ablation, sputtering and milling, chemical, sodium borohydride, potassium bitartrate, methoxy polyethylene glycol, and hydrazine, and the last is the biological, such as plants and microorganisms. But according to the hazardous substance fact sheet, sodium borohydride, it can cause irritation and burn, shortness of breath, etc. So it basically indicates the toxicity or highly hazardous usage of a chemical. So in my case, the biological method is more preferable. So I'm using plants instead of microorganisms because it has been reported that the plants can have a quicker reaction as compared to microorganisms. So based on the problem statements, limitations of conventional anti-cancer therapy, as well as the hazardous chemicals used for the synthesis of silver nanoparticles, we evaluated the anti-cancer activity of silver nanoparticles synthesized by this plant on two models of cancer cell lines, which were JCAT, human leukemic T cells, as well as the HT29 cells, which is human colorectal cancer cells. So the specific objective were to examine the effects of the silver nanoparticles on the proliferation of JCAT and HT29 cells to evaluate early detection of apoptosis in JCAT and HT29 cells treated with these nanoparticles, and the third one to analyze the cell cycle of JCAT and HT29 cells treated with these nanoparticles. So this is the methodology that we applied. Basically, we used silver nanoparticles that consist of 10% of catharanthus roseus aqueous extract in 5 millimolar of argentum nitride. And then this is the cells. So after the treatment, we use these assays to evaluate the anti-cancer activity, which were MTS assay, the viability assays. So we use concentrations ranging from 1.96 to 1,000 microgram per ml in double dilution manner, incubation times 24, 48, and 72 hours. For the annexin concentrations, we just use the selected one, 5, 10, 15. Incubation times, we added one earlier incubation times because of these assays is specifically to detect, is quite specific to detect the early onset of apoptosis, and followed by 24, 48, and 72 hours. For the cell cycle, we just use 10 microgram per ml, incubation times 24, 48, and 72 hours. And after that, statistical analysis, which was the one we went over. So this is some information about the characterization of the nanoparticles. So moving on to the results and discussion, this is the MTS assay results. Proliferative effects of the silver nanoparticles on JCAT cells. So basically, the silver nanoparticles have inhibited the proliferation of the JCAT cells, especially at the concentration of 7.82 until 500 microgram per ml at all incubation times. On the other hand, we can see there is an induction of the proliferation of the cells at a very low concentration, which was 1.96 microgram per ml after 72 hours treatment. So the median IC50 value was 5.87 microgram per mil. So in comparison, when we compare the effects with aqueous extract, we did not observe any induction of the proliferation, but still the significant inhibition of proliferation was observed at most of the concentrations. And the median IC50 value was 361.72 microgram per mil. So this is the results for the proliferative effects of the silver nanoparticles on HT29 cells. Just now, Jacat, this is HT29 cells. So we can see a similar trend whereby there is inhibition of the cell proliferation, 15.6 until 1000 microgram per mil at all incubation times, as well as induction of the proliferation of the cells, which were at low concentrations, 3.91 and 7.82 microgram per mil. And the median IC50 value was 13.19 microgram per mil. So in comparison, when we use catharanthus roseus aqueous extract, we did not see any induction of cell proliferation, but only the significant inhibition of the cell proliferation. And the median IC50 value was 419.26 microgram per mil. So we can say that from the median IC50 values, the order of the cytotoxicity was, the highest was by catharanthus roseus silver nanoparticles on Jacat cells, followed by silver nanoparticles on HT29 cells, and then aqueous extract on Jacat cells, and the last is the aqueous extract on HT29 cells. So we can say that the silver nanoparticles has produced higher cytotoxicity effects than the aqueous extract. So it might be due to the different phytochemicals responsible in the synthesis of the silver nanoparticles that have contributed to the cytotoxic effects on cells, and probably the compounds involved, the fatty acids, esters, and alkaloids. This is based on the literature. So the cytotoxicity effects also can be due to the unique features of the nanoparticles itself, such as the small size, high surface area to volume ratio, surface functionalization, which will lead to the biokinetics of the silver nanoparticles and increase the cytotoxicity. So previous study has reported on the higher number of nanoparticles were observed in mouse fibroblasts compared to silver microparticles that lead to the induction of reactive oxygen species, DNA damage, and eventually apoptosis. So the second point from the MTS assay is about the Jacat cells that were more sensitive compared to HT29 cells, either in response to the nanoparticles or aqueous extract. So the anti-cancer vinca alkaloids present in the catharanthus roseus are commercial anti-cancer chemotherapeutic drugs to combat acute lymphoblastic leukemia, and they are namely the vinblastine and vincristine. So probably these compounds are actually present in the nanoparticles or the aqueous extract that that produce higher cytotoxicity effects as compared to HT29 cells. Other possible anti-cancer alkaloids include vendacin, venipidine, and vincrosidine, which are antitubulin properties that have antitubulin properties, may inhibit the formation of mitotic spindles by damaging microtubules and will eventually cause the cell cycle arrest. While for the induction that was observed at low concentrations of silver nanoparticles on these two cell lines, this is a differential effect whereby a high dose increases cytotoxicity while low dose induces cell proliferation. And there is a term suggested which is called as hormesis, which can be defined as a potentially toxic agents cause stimulation in lower doses. The crude plant extract used in the nanoparticle synthesis contains numerous active compounds that interacting to one another, so probably this might be the reason why there is an induction of the cell proliferation. apart from the anti-cancer compounds, non-enzymatic antioxidant molecules such as ascorbic acid, alpha-tocopherol, reduced glutathione, and antioxidation enzymes have been reported can scavenge the reactive oxygen species. So this is the results, the second one, which is the expression of the externalized phosphatidylserine on jacket cells treated with the nanoparticles. So basically, we use annexin conjugated with FIT-C and in conjunction with propidium iodide to differentiate between early and late apoptosis. For each concentration, the first one from left is the annexin negative PI, PI positive, meaning this is for the necrotic cells. The second one is for the late apoptotic cells, both positive, annexin positive, PI positive. This is for the viable cells, both negative. And the last one is the early apoptotic cells, annexin positive, PI negative. So for this result, we observe that the silver nanoparticles have produced a higher number of late apoptotic cells as compared to early apoptotic cells. So basically, we're just comparing the second one with the fourth one. This is the results on jacket cells treated with aqueous extract. And we can see that for 6 and 24 hours, early apoptotic cells were higher compared to the late apoptotic cells. But in the following incubations, which were 48 and 72 hours, the late apoptotic cells were higher as compared to early apoptotic cells. And this is the HT29 cells treated with silver nanoparticles. At 6 hours, the late apoptotic cells increased, but when it comes to 24, 48, and 72 hours, the early apoptotic cells were actually higher as compared to late. And this is the HT29 cells treated with catharanthus roseus aqueous extract. So the significant difference were observed at the first incubations, 6 hours and 24 hours, whereby the early apoptotic cells were higher as compared to the late apoptotic cells. So at 6 hours, catharanthus roseus aqueous extract produced higher percentages of early apoptotic cells, while catharanthus roseus silver nanoparticles produce higher percentages of late apoptotic cells. So this indicates that the aqueous extract started to induce apoptosis after 6 hours, and silver nanoparticles induce apoptosis earlier than 6 hours. So this summary is basically we have calculated the difference, because just now I just explained, particularly specific within the graph, that when we compare among the treatment, so this is basically the summary that we have. So at 24 hours, the results was as correlated with our MTS-assay, which is in order as what we observe in MTS-assay. But at 48 and 72 hours, there is exchange of the treatment, whereby instead of the nanoparticles here, aqueous extract on jacket cells were shown to have higher percentages of both early and late apoptotic cells as compared to the nanoparticles on the HT29 cells. So this might be due to if we look at the MTS-assay back, the induction of the cell proliferation was observed at low concentrations, which were 3.91 and 7.82 at 48 hours, and 1.96 and 3.91 microgram per mil at 72 hours. So these concentrations are within the ranges of the concentrations used for annexin PI staining, and probably this could be one of the reasons why the apoptosis, there's a changes in the apoptosis, in the percentages of the apoptotic cells. Okay, this is the results for the cell cycle assay. treatment effects of nanoparticles and aqueous extract on the cell cycle of JCAT cells. And here, for each histogram from the bottom, this is for the G0G1 phase of the cell cycle, this is for the S phase, and this is for the G2M phase. So we can see that upon the treatment of aqueous extract and nanoparticles, it has arrested the S phase at all incubation times. Whereby this is the results on the cell cycle of HT29 cells. So it is not the same like JCAT. At 24 hours, it basically arrested G0G1 phase of the cell cycle. And then when it comes to 48 hours, aqueous extract has arrested S phase, while the nanoparticles arrested the G0G1 phase. And at 72 hours, the aqueous extract arrested the S phase, and the nanoparticles arrested both S phase arrest. So we can see that this is basically the conclusion for that. The nanoparticles on JCAT cells arrest S phase at all incubation. Nanoparticles on HT29 cells, G0G1 as well as S phase. Aqueous extract on JCAT cells, S phase at all incubations. And aqueous extract on HT29 cells, G0G1 phase at 24 hours, S phase at 48 and 72 hours. The DNA damage may arrest or suspend the cells in either G1, S, or G2 before undergoing apoptosis in case the damage cannot be fixed. So oxidative stress activates the p33-MAPK signaling pathway and inflammation of the transcription factors. These are the probable mechanisms that have been reported, or it can also affect the mitochondrial dependent N-terminal kinase pathway, which will cause a disruption of mitochondrial respiratory chain, increase the reactive oxygen species, and interferes the ATP synthesis, which eventually damage the cellular DNA. These are the possible phytochemicals involved in the plant itself that are responsible for the cell cycle arrest. For example, here is the vinca alkaloids that have antitubulin properties, disrupt and interfere microtubules, and these will cause the M phase arrest. Vincristine has been reported can cause the G2M phase arrest, and probably because of we did not find the arrest in this phase, so the arrest probably due to other compounds in the plant. The camptothecin used has arrested S and G2M phases, which is as according to the many previous studies, but AGNO3 used in this study has arrested G0G1 phase at 48 and 72 hours in HT29 cells. So this requires further investigation since it did not inhibit any the proliferative activity in the MTSAC for the AGNO3. So for the conclusion, we have correlated the proliferative activity, induction of apoptosis, and the cell cycle arrest. So JCAT and HT29 cells have undergone severe nanoparticles induced stress. However, further analysis on the detailed mechanism of cytotoxicity and cellular uptake are required for better understanding on the cellular interaction. Because the major drawback associated with a new drug development include lack of specificity and uncertainty with its cytotoxicity on normal cells. So, we plan to do some further analysis on the normal cells counterpart. Besides, in vivo studies are necessary to address the formulation of biogenic silver nanoparticles as an alternative to conventional anti-cancer drugs. Experimental evidence indicating that the silver nanoparticles have been shown to induce higher cytotoxic effects compared to the aqueous extract, and this due to the small size themselves that have increased its effectiveness to penetrate cells, which will lead to the cell death. So this research has expanded the knowledge on the comparison cytotoxic effects between nanoparticles as well as the aqueous extract on these two cancer cell lines. So it provides a very strong foundation to develop better strategy of cancer therapeutic agents. So these are the references used, acknowledgement, science fund grant, as well as my university's fund. These are the co-researchers involved in my study. Thank you for your attention.